Okay. Okay. All right, everybody, so let's get started. Welcome to the final seminar of the quarter for entomology. We're ending with a bang today with Dr. Jeffrey Fader. He got his PhD at Michigan State University working with Guy Bush. He then had postdocs at Princeton and University of Chicago. Following that, he got a faculty position at the University of Notre Dame, where he is currently a professor in biological sciences. His lab there works on speciation and host specialization in the Rhagoletis pomonella species sibling complex. And they use field work and selection experiments and molecular genetics to answer all sorts of interesting questions in this area. They also are looking at color polymorphisms in a bioluminescent Jamaican click beetle. He's also the director of the GLOBES program there, the Global Linkages of Biology, Environment, and Society. So without further ado, I'll get started.
Oh, thanks. Can everybody hear me in the back? It's fine. Good. Thanks, Ian. Thanks, Meredith, for inviting me. It's been a lot of fun so far. Thanks, you all, for being here today. And I'm going to tell a story about genomic architecture of speciation today. And of course, speciation is the ultimate source of biodiversity, and so this is sort of the big issue, at least in putting the talk today in the context. We're trying to understand speciation and biodiversity, but specifically I want to sort of concentrate on the question of what's the nature and role that genome structure itself might play in speciation. And I'm particularly interested in this question with respect to taxa or populations that are diverging in the face of gene flow or migration between the populations. That is, let's say, in a geographic context of what we'd call parapatry, partially geographic overlap of populations, or sympatry, that is, would be more complete geographic overlap of populations. Now, we've learned quite a bit about the genetics of speciation over the last couple of decades. And particularly with sort of an emphasis on identifying individual genes that are contributing to reproductive isolation. We could call this sort of the hunt for speciation genes. And this has been quite informative about telling us about the genetic basis of reproductive isolation that can be contributing to speciation. There's a couple of difficulties here, though. Many of these genes that have been identified so far affect post-zygotic incompatibilities, and a lot of times these crosses are done to investigate the genetics of speciation through sort of long established and somewhat highly diverged taxa. And so some of these loci may have actually diverged after speciation was essentially complete. And also, more importantly for the talk today, the hunt for the speciation genes, it's been quite informative, but it also misses another issue, and that is thinking about how the genes themselves that are diverging are arrayed together in genomes and might diverge collectively and might influence further differentiation during the formative stages of speciation. And so today we're going to concentrate on this fly here as our main character, Rhagoletis pomonella. Rhagoletis pomonella, of course, is a model system for ecological speciation with gene flow in phytophagous or host-specific phytophagous plant-eating insects. The basic premise for Rhagoletis pomonella is that in the process of shifting and adapting ecologically to new host plants, this provides an impetus or some of the main factors that are driving the divergence process. And the particular example of Rhagoletis pomonella shifting from its ancestral host, hawthorn or thorn apples, to introduce domesticated apples about 150 years ago in the eastern United States is often cited as an example of this type of sympatric or ecological speciation with gene flow in action in our own backyards. So this provides us with at least a known geography, sympatric, geographically overlapping, and a historical time frame, then about the last 150 years ago the shift occurred, that allows us to sort of investigate, or sets up the preconditions at least for doing an investigation of genomic patterns of differentiation underlying speciation with gene flow in the very early incipient stages. And the take-home message I want to impart on you today, what we're going to do is we're going to integrate geographic surveys of genetic differentiation across sites with selection experiments and mapping experiments. And the take-home message is that surprisingly, or somewhat unexpectedly, at least in my opinion, what we seem to find are large continents of differentiation across the genome rather than small sort of isolated islands or genomic islands of speciation. A different genomic architecture, at least underlying the incipient stages of host race formation in this fly than I would have thought. And what we're going to, one of the sort of implications of this is that maybe these, and I'll explain this a little bit more fully as we go, some of the low baseline levels of differentiation that we detect in genome scans. And by that, a genome scan, what I mean is looking at populations and scanning a number of loci across the genome of the fly or organisms to characterize genetic differentiation on a sort of more genome-wide scale. Many of these studies sort of set a baseline level above what we'd expect statistically for neutrality. In many cases, actually, selection may be more genome-wide and widespread, right? And that low level of neutrality that we set for statistical purposes may actually be too high, and we're missing a number of regions of the genome might not be under as strong a selection, but are still under divergent selection during these early stages of population divergence. And that's the take-home message that I want to impart, and that's what's shown in this figure here. Rather than this view of sort of genomic architecture of divergence in these host races of Rhagoletis flies, we see something that looks more akin to sort of a large uplifting and more widespread differentiation. Okay, so let's sort of take a step back and sort of lay what I'm talking about today in a larger context. And that I'm saying, well, what often initiates speciation? And many times it could be that reproductive isolation due to divergent selection, adapting populations to different habitats, is an initial impetus or driver of the differentiation process. And so what I'm saying here then is that you could think about it in the context, let's say, of fitness trade-offs. Maybe we have two habitats, and you can see the association here with apple versus hawthorn for Rhagoletis pomonella, the host that attack. Maybe we have two habitats here. One environment, like a desert environment. The other environment, maybe an alpine meadow environment, right? And what we've got is differential selection pressures on traits to adapt the population to those different habitats, such that maybe a gene or an allele that's favored in producing a phenotype that confers higher fitness, let's say, in our desert habitat, has the opposite effect and results in a phenotype of lower fitness in the alternative habitat, and vice versa. So this is our context for divergent ecological selection. And if this is the case, then we'd expect sort of to see a heterogeneous pattern of genetic differentiation throughout the genome. So what do I mean by that? Well, what I mean is that for genes that are directly under divergent selection between the two habitats, producing a phenotype that's directly selected for differentially, we'd expect to see if there's some gene flow going on between the populations. For these traits that are directly the targets of selection or nucleotide sites, we'd expect to see strong differentiation between our populations. So these regions of the genome, if we were to scan through, should show high levels of difference between our populations. We could also have genes that aren't directly under selection, or sites that aren't directly under selection, but are physically linked close by. And those sites should also show a degree of differentiation between our populations, but not as strong as those sites that are directly under selection. And then finally, we should see sites that aren't directly under selection, again, neutral, at least in terms of fitness effects. but unlinked, that is, at greater recombination distances to a locus directly under selection. And in this case, these genes should flow more freely between our populations that are divergently selected, and we don't expect then to see high levels of genetic difference between our populations if we were to score individuals at this locus in genotype level. So far, so good? Okay, so we expect a heterogeneous pattern of genomic differentiation, which sort of has led to a genomics islands of speciation sort of analogy. And that is, we might expect to see, initially during the speciation process of speciation with gene flow, certain regions, just a few, scattered through the genome that are under high divergent selection that show differentiation between our populations. And then by a process that Sarabia has called divergence hitchhiking, we'd expect to see then reduced levels of gene flow in the immediate surrounding regions of the genome for those sites that are directly under selection. And this could allow then the accumulation of additional new mutations that have smaller selection coefficients for them, actually, to hitch a ride, and could facilitate then the fixation or the differentiation of these new mutations. And so we see a buildup of additional divergence around these initial sort of bridgeheads or beachheads of divergence for strongly selected sites. And this has been argued to be a possible process that facilitates divergence with gene flow speciation. So here's sort of another view to sort of think about this island analogy, this genomic islands of divergence analogy. What we have here, what I show here is the extent of genetic divergence, right? And here we have like our seafloor, which would be really a truly, purely, neutrally evolving region of the genome. And then what I have here is our sea level. This would be sort of statistically what we position as the level above which we could statistically say we have differentiation between our populations above neutral expectations. Therefore, either the site is linked to or actually under selection. So here's our island popping above the sea level. Here we have our selected locus, and here we have surrounding sites that are relatively, that could be tightly linked or linked to our site under selection that could also rise above neutrality, at least neutral expectations of the amount of divergence between our populations. And what we could then do is a genome scan. Right? And what I have here is FST, degree of FST, which is a measure, a summary statistic of the degree of divergence between populations. And what one could do then is look at a number of loci distributed across the genome, and then set for all the loci a level. There is our sea level then of neutral expectation, above which statistically we could say we have evidence potentially for divergent selection acting on this region or this locus or near this locus in the genome. So down here we have all of our so what we call non-outlier loci that are putatively neutral, and then the whole genome of our population pairs that we might be comparing. We could have certain outlier loci that statistically look to be more diverged than they should be based on neutral expectations. And we could say that those outlier loci are putatively under selection and might be candidate loci for the targets of divergent selection or closely linked to them. And so one of the classic studies that sort of established this sort of speciation island type of thinking or analogy was done by old native son Tom Turner from Davis, and looking at mosquitoes, Anopheles gambia, the M and S forms. And they did a systematic scan by looking mostly at microarray hybridization and characterizing levels of divergence between these two forms of mosquitoes. And the take-home message was that they really only found just a few levels of the genome, or regions of the genome and markers that exceeded this sort of threshold sea level. And they christened the term genomic islands of divergence to characterize these few regions. Now, subsequently, there have been a number of genome scans that have been conducted in animals and plants, and there's been some generalities that sort of emerged from these studies that outlier type loci typically comprise a small portion of the genome, roughly 5 to 10%, with a mean of at least 8.5% in animals. There's been recent compilation of data from plants as well. It seems to be sort of a remarkably consistent pattern. I think it's 8.8% or something that have been discovered in a number of genome scans of plants. Some of these studies have mapped some of these outlier loci, and in some cases the loci appear to be clustered within specific and isolated genomic regions. In other cases, they're not. So there's no really consistent pattern with respect to loci clustering. In some cases, QTLs have actually been associated with some of these genomic regions during differentiation for specific traits, and in other cases, not. But there's an alternative to this idea of divergence hitchhiking as well. And we could characterize it as an idea that Bill Rice first talked about in the 1980s, and in this case, we could characterize it as sort of widespread multifarious selection. There's another possibility. And here, the situation may be that actually strong selection is working on a number of different regions of the genome initially during the divergence process with gene flow. That is, it may be that in often cases where we see differentiation take place in the face of genome, it may really require initially strong selection at a number of different places in the genome. And in this case, the effective level of migration between populations may be sufficiently reduced genome-wide, rather than just in specific regions, to allow the subsequent accumulation of new mutations, irregardless of linkage, across the genome. Essentially, then, gene flow would become reduced sufficiently genome-wide that these populations are approaching what would be the situation if they were allopatric, geographically separated, and not exchanging genes at all. And so therefore we could see sort of the rising boat across the whole genome and not just in isolated regions. At least that's an argument. Okay, so now we have a case study, Rhagoletis pomonella. We can examine these alternatives. Here's a case of incipient sympatric differentiation associated with the host shift, and the premise being adaptation of the ancestral hawthorn-attacking population of these flies, shifting hawthorn populations, shifting over to apples, ecologically adapting, and those ecological adaptations producing some reproductive isolation. And we could look at the pattern of differentiation genetically across the genome of these flies and see if it fits more with like this island view, or if it fits more with a sort of widespread multivarious view of selection, divergent selection. And so what I'm going to tell you now is I'm going to couple some population surveys of these flies with some selection experiments on specific phenotypes that we know and environmental factors that we know are contributing to ecological adaptation in these flies, in particular diapods or life history related traits, and along with mapping studies. So first let's look at some of these population genome scans, in which we've looked at, in this case, 33 microsatellites and 6 allozymes to get a picture of the view of genomic differentiation in these flies. And what I'm showing here is a map of our collecting sites. We've got 4 different sort of co-occurring populations that we surveyed that have apple infesting populations and hawthorn infesting populations in close geographic proximity, within like 100 meters or so, arrayed in a latitudinal distribution across the Midwest. And I'm also going to look at one population of hawthorn infesting flies from Texas. This just shows you the range of the apple and hawthorn races of Rhagoletis pomonella. The red is the apple race. It's mostly in the eastern and midwestern United States. And the hawthorn infesting fly extends down into the southern United States. And here's a map of the location of the markers that were investigated. You can see that they're scattered throughout the genome. There's actually 6 chromosomes in this fly. There's a small dot, 6th chromosome, that we don't have any markers on. But generally speaking, the markers are just arrayed across or throughout the genome of these flies. There's no single linear map arrangement for some of these markers, as you'll see here. They form sort of complex patterns, and that's because there's inversion polymorphisms in several of the chromosomes of these flies. So that there's no single universal map order in all individuals. So here's the result of the outlier analysis looking at population differentiation across these 4 sympatric pairs in this population from Texas. And what we see from this, and here's our FST values, and here's our statistical cutoff then for neutrality. And the take-home message from this is that we actually see a pattern that's really consistent or seems to support the island hypothesis. There's actually only 3 loci in 2 genomic regions in this fly that showed significant outlier status. However, on closer examination of the data, particularly looking more specifically at the overall patterns we see across the genome or across geographic collecting sites, it's a much more complicated pattern, and there's evidence for widespread differentiation across the genome in these flies. And this really highlights why it is important to actually not just to confine yourself to a pair of populations, but really to get the whole geographic mosaic of population differentiation when one's trying to take into account how much of the genome is actually diverging with speciation and gene flow. And what I show here is just 8 of the loci. This is a general pattern that all 39 of the loci that we looked at showed. Just basically every single locus, at least in Hawthorne populations, Hawthorne-infested populations, you can see allele frequency here. Here's our latitude of sites. The black here are our Hawthorne sites. And what you see is that basically every single locus showed latitudinal declines within the Hawthorne race. In comparison, the apple race, most of the loci also showed latitudinal declines in the apple infesting race. Sometimes that climb was in a similar direction as the Hawthorne race, as shown here and here. Sometimes the slope of decline in the apple infesting population was shallower than that in the Hawthorne infesting populations, and sometimes it was steeper. And in other cases, we actually see a reversal of declines themselves in the apple. The sign of decline or directionality of decline was opposite. And so basically, we found evidence for significant differences between apple and Hawthorne in their allele frequencies as microsatellites and allozymes at 32 of the 39 markers we surveyed, the majority. And the patterns of linkage disequilibrium and the mapping of these markers indicated that the loci are really distributed throughout the genome. And also that the 32 loci represent at least a minimum of 17 different genomic regions of this fly genome showing host-associated differentiation. A much different pattern than one got from looking at the outlier analysis.
Jeff, do you know what the karyotypes are in these populations with the inversion?
I wish I could. The problem with frugivorous tephridids is they have lousy polytene chromosomes, which has always been a hang-up. So we've always had to go back to the old-fashioned roll-up-your-sleeves stir-event way of just mapping and looking at recombination distances amongst crosses to infer inversion polymorphism. Now, hopefully, we're beginning to do a lot of high-throughput sequencing, so we may, in fact, from specific crosses, eventually maybe we'll get lucky and find some of the breakpoints in some of these crosses and be able to begin to map inversions. And I think there's overlapping inversions, polymorphisms in these populations, and eventually maybe we'll be able to do that. But right now, we still have to rely on good old-fashioned genetic crosses and variation in recombination rates between crosses to infer inversions.
So in the obvious interpretation, you're just seeing latitudinal declines in inversion frequencies? There's loads of loci in there that are associated with
Exactly. And it turns out this is another aspect of the story. Some of that inversion polymorphism probably was introduced by introgression down from up from Mexico. So that's exactly what we think is going on. That past periods of geographic isolation contributed to inversion differences between the populations. Subsequently, there was some contact and introgression in which adaptive clines formed in the ancestral hawthorn infesting population, and the variation in life history in those clines contributed to this fly being able to explore new host plants locally and sympatrically that had different fruiting times and required different life histories to adapt to. Exactly. That makes sense to everybody? Okay. So, I hope. So let's look at the life history of this fly, and let's look for factors that might be causing genome-wide adaptation or genome-wide selection. And here's a diagram here of the natural history and biology of Rhagoletis pomonella. And we'll sort of work this in now to, once we understand the biology more, to see how we can look at certain aspects of the diapause life history that might be under strong divergent selection between apple and hawthorn flies. And then we'll try to link selection occurring or associated with those traits, those life history traits, with the pattern of genome-wide differentiation. Okay, so Rhagoletis is univoltine. It has one generation per year. It overwinters in a facultative pupal diapause. And in the summer, the flies close and emerge as adults. They then rendezvous on their respective host plants to mate and oviposit. Here's their mating. Here's the female ovipositing the fruits. They lay eggs, usually singly in a fruit. The eggs hatch. Larvae feed within the fruits. That's why they're major economic pests. And then when the fruit abscises from the trees in the fall, the larvae then leave the fruit, they burrow into the soil an inch or two deep, and they pupate, completing the life cycle. So a key consideration is that apples and hawthorns fruit at different times of the year. They have different seasonalities. In fact, preferred apple varieties fruit about 3 to 4 weeks earlier than hawthorns in the field season. And therefore, ripe host fruits that the females oviposit into and flies use as rendezvous sites for courting and mating actually represent seasonally different resource islands. That the fly, since it's univoltine and has only one generation a year, has a longevity of about a month out in nature, they have to match their life histories to maximize the availability of suitable host fruits. And so you can imagine now that selection pressures are different on apple and hawthorn flies in order to achieve the synchrony between their life cycle and host food availability, their temporal resource islands. And how do they do that? Well, there's two primarily areas of divergent selection probably on the life history of these flies. One is, if you think about it, the flies have to hit a target when they're coming out and closing as adults. And that apple target is earlier in the season than the hawthorn target. Consequently, apple flies have to break diapause sooner and complete development earlier than hawthorn flies in order to utilize the earlier seasonal fruiting apple host. And here's some results from sort of standardized or garden type experiment in which apple and hawthorn pupae were reared under the same environmental conditions. These are two sites in Urbana, Illinois, and Grant, Michigan, and the basic idea here is just to show you the mean number of days to eclosion for apple versus hawthorn adults. And what you can see here is that apple flies do in fact eclose under standardized conditions about 2 weeks earlier than hawthorn flies, fitting the chronology of when their host plants fruit out in nature. And you can also see that there's a bit of clinal variation in this characteristic and trait as well, and flies from the north actually eclose a little bit earlier than flies from the south. Conditions are colder earlier in the season in the north, and so flies have to actually develop faster to hit their targets up north than in the south. So that's one aspect of the diapause life history. That's under differential selection. But diapause isn't a single trait. And another trait, if we think about it, if we shift our attention from a timing of adult eclosion to what's going on earlier in the pupal life stages of these flies before winter rather than after winter, there's also another window in which divergent selection is affecting diapause life history. And what is this? Well, if you think about it, apples abscise from trees earlier, also before the onset of winter, than hawthorns do. And so consequently, apple larvae leave apple fruits and pupae earlier in the season relative to the onset of winter than hawthorn pupae do. Therefore, there's a longer period of time that apple fly pupae has to remain in the ground before the onset of cold winter temperatures than is the case for hawthorn pupae. And it turns out that Rhagoletis are facultative diaposers. That is, if they're exposed to warm enough conditions for a long enough period of time, they'll forego a deep pupal diapause and they'll begin to immediately develop into adults, which is disastrous out in the field because they will close at a time in the fall when suitable host fruit isn't available. So there's stronger selection then, at least in for the earlier phenology of apples, to select for a deeper initial diapause for apple flies to avoid non-diapause development. And so here's some similar carbon garden standardized environmental rearing experiments with apple and hawthorn pupae, in this case along from Urbana, Illinois, up again to our Grant, Michigan site. And what we see here is the percentage of non-diaposing pupae in the population. And you can see that at all sympatric sites, there's a lower percentage of non-diaposing that you can use in apple flies. Earlier fruiting, they have to withstand hotter temperatures for a long period of time than the corresponding hawthorn population at the sympatric site. And again, you can see clines. So hawthorn flies have to develop relatively more rapidly and push the limits to get in before winter hits. Therefore, they're more susceptible to non-diaposing development than apple flies are. Okay, so what does this allow us to do then? Well, this allows us to test for genetic associations with diapause life history in these flies. And we could look, for instance, for gene frequency correlations at some of the markers that we said before showed differences between the populations. We could look for associations with the timing of diapause, adult eclosion time in these flies, right? And we could also actually conduct selection experiments, for instance, by lengthening the pre-wintering period for hawthorn flies, right here. Normally, let's say they experience about 7 days of room temperature before the onset of winter. That would be standard conditions that a hawthorn fly might experience, let's say, at our Grant site. Right? But we could lengthen that. We could make it, let's say, 32 days. And by doing so, by increasing that pre-wintering period for hawthorn flies, we could essentially create conditions that pupae would face if it was infesting the alternative apple fruit. And then we could look for a genetic response in these selected, different selected conditions and try to match it to the patterns that we see geographically out in nature between the populations of apple and hawthorn flies that show differences across the genome. So do they differ? And the answer is, and here's just a summary slide, this is the results for the selection experiment, yes. In fact, of the 17 genomic regions that I said before that show host differentiation in the population survey, turns out 16 of them showed significant response either in the selection experiment or related to adult eclosion time. 26 of these loci total responded in the pre-winter selection experiment. They represented at least 16 different regions. With respect to correlations with adult eclosion time, there are 20 loci that were significant, representing at least 10 different regions of the genome, and 10 were shared between the selection experiments and eclosion time studies. we could explain almost 50% R squared of the variation in occlusion times just based on these microsatellite markers. So it would appear that by connecting these selection experiments, at least with the pattern of genomic differentiation in nature, we can rule out at least patterns that were generated by drift or isolation by distance, and have some confirmation that, in fact, genome-wide selection on life history, diapause life history, could result in widespread patterns of genomic differentiation across the genome of these flies. So we can connect some of the dots and say that we can make some sense of seeing a continence pattern rather than this alternative of genome island view of differentiation in Rhagoletis. Now, this is a recent study that Nora Pazanski's lab just completed of high throughput sequencing of Anopheles gambia mosquitoes, the M&S forms. Remember, this is the initial study system that really captured the imagination of genetic architecture students in terms of thinking of this genomic islands view. And there were 3 regions that in an initial Turner study that were shown to show high outlier status as evidence of genomic islands of speciation. However, in a more thorough high throughput sequencing, it turns out that this view is mistaken. And now looking at the M&S forms, it turns out that there's evidence for widespread genomic differentiation between the M&S forms of Anopheles as well. So Rhagoletis may not be alone. The classic study system that sort of really captivated our imaginations about genomic islands of speciation turns out to more fit more of a, at least an archipelago view of differentiation through the genome. Okay, so what about the theory behind islands of genomic divergence? Why were we thinking so strongly that this view should mostly characterize early patterns of ecological differentiation with gene flow? And part of the results or reason for this thinking came from some theory that Brian Charlesworth did and colleagues, although it was not directed to the question of speciation with gene flow, but it was used to make an argument for why divergence hitchhiking could really facilitate sympatric speciation or speciation with gene flow. What Brian showed was essentially trying to understand background selection in the context of a metapopulation with local population adaptation. But this figure that he generated was used to make an argument for divergence hitchhiking. And what I have here, and what we're showing here, is a figure where Brian and colleagues estimated what the FST value, the degree of differentiation, should be between populations. For a site under strong selection, here's a site under strong selection, and then we could figure out what the expectation should be at equilibrium for a neutral site at various recombination distances from that strongly selected other locus, right? And this is the pattern that Brian and colleagues generated, and the key point here is that even at recombination distances that are quite far from the selected site, there should be a significant amount of FST, of genetic differentiation for neutral sites. Therefore, when we scan the genome, we should frequently pick up, even at rather far mapping distances from a selected site, evidence for neutral differentiation. At least that was the argument. And here we see what the value should look like for the selected site being under a lower selection coefficient of 0.1. It's still fairly pronounced. It may only go out to be like 5 centimorgans, but we should still see it. But what I want to draw your attention to is a couple things. One, Brian didn't intend, I think, for this simulation and analytical study to be used for analysis of speciation. 2 things. One is that his effective population sizes, this is really a local adaptation model. His effective population sizes that were used were quite small, 1,000 individuals in each of these small isolates. And also the migration rate is very low between the local populations, 0.001. So what happens when we actually use more realistic parameters for migration rate and population sizes? What's the expectation at least at selection, migration, drift equilibrium for FST between our populations? And here's the original results of Brian. And here's what we've been estimated. In fact, if we just increase the population size up to 100,000 perhaps, and move the migration rate up just a little bit to 1% per generation. And what one can see is that that island of neutral differentiation, at least at equilibrium, quickly goes to 0. So under more realistic parameters, at least, we might not, for speciation, we might not expect to see large islands of neutral differentiation surrounding a strongly selected size. There it is. We've subsequently derived what the estimates should be for the establishment of a new mutation. And they produce very similar results. I don't have time to talk about it in the talk right now, but in the questions and answers, if you're interested in looking at what those results are, I could show you. But the take-home message is pretty much the same. That under more realistic parameters, at least for selection, migration rates, and population sizes, we don't expect to see tremendously large islands of neutral differentiation or tremendously higher probabilities of fixation or establishment of new mutations around a selected site. We could increase the number of loci under selection in these models and see what happens. And in this case, the results really say that there could be 3 phases of speciation resulting. That when there's just a few number of loci, in this case up to 4 loci under strong selection, here we've got a migration rate of 0.1, effective population size of 100,000, and we're adding more loci under strong selection to the genome and seeing what happens. What we see is that the same pattern holds up until maybe you get 4 strongly selected, 5 strongly selected loci. And then what you begin to see is a period of time after this initial divergence between our populations where you can get a little bit of an effect of facilitation of divergence hitchhiking. Right? So you get an elevation around our selected site. And then after you get to about 10 or 11 loci, we see the results of what we could call genomic hitchhiking. That the level of effective gene flow is reduced enough that we begin to accumulate neutral differentiation across the genome. So you go through some of these phases, at least, with speciation with gene flow, where initially you get a period where there's a number of different loci that have to be fixed. You don't see much genomic hitchhiking. Then you go through an intermediate region, a middle region, where you could see evidence for some facilitation of genomic hitchhiking, although the important point here is that it still has to be, the site still has to be fairly close to the other locus under divergent selection. It still needs to be a couple of centimorgans or so. It's not as widespread. It's not 10 centimorgans. It's not 5 centimorgans. It's still rather restricted. And then you go through a later phase, at least, where there's enough loci under divergent selection that differences accumulate all through the genome.
So each locus you have there is just something like unlinked? Like
exactly. Each locus is unlinked, so we're adding unlinked loci, and then we're saying, in this case, let's get a new mutation or look at the accumulation of neutral differentiation for a site at a given recombination distance to one of those loci in the genome that's under strong selection. Yeah. And this is the general pattern you'd expect to see sort of phases that the population goes through. There is an effect that's contributed by divergence hitchhiking, but it's not as strong as we might have expected, and you still have to have relatively tight linkage of those sites to the other site under strong, one of the sites under strong selection.
How big is the genome?
Of Rhagoletis?
No, of this thing.
Of this thing, we can make it infinite because we're just adding new sites that are new sites that are unlinked. But that it does raise an issue that genome structure, the number of chromosomes, the recombination rate, and how they're distributed could influence the probabilities or effects that genomic hitchhiking, divergence hitchhiking could have.
So every time you add a new selected locus, you add a new chromosome.
Essentially, yeah. You could think of it that way. Yeah. Yes? Okay, so in the last part here, I just want to try to make a connection, and I'm off camera for a second. I shall return. That could we find at least other evidence from the transcriptome of the fly that allows us to tie together the idea that widespread selection on diapause-related life history traits could generate widespread divergence between these flies and their genomes. Could we make that connection at least even more tangibly? And so what I want to show you now is moving more towards the physiological and genetic basis for diapause traits through this type of expression analysis through the life history of these flies. And so what we've been doing is working on the genetics of diapause phenotype by characterizing the transcriptome of Rhagoletis pomonella through larval pupal stages and different pupal stages and adult stages. This is work done with you, Robinson, Stuart Berlacher, Dan Hahn, Greg Ragland, and Dietmar Schwartz. And we've been also looking at sort of the genomics of dormancy phenotypes by characterizing sort of physiological landmarks through diapause time course. And we can do this taking a respirometer approach, measuring metabolic rates through different life stages of the fly, essentially going as they go into diapause and they go out of diapause, to look at differential expression patterns of marker loci. And just to show you we're interested in diapause initiation again as one key area of divergent selection on these flies, as well as diapause termination or breakage. Those are two critical areas or windows that we know or have good evidence that divergent selection is acting between apple and hawthorn flies. And this is just to show you, as we go into diapause, we can now take individual pupae and measure their respirometry rates, or CO2 production, to characterize the diapause characteristics, at least, the tendency of that individual pupae. And what we see here is that there really is a difference between those individuals that go into diapause and are true deep diapausers, as opposed to those that are non-diapausing. Diapausing flies really dive into a really reduced level of metabolism, whereas non-diapausing flies, turns out they decrease metabolic activity, but they don't dive into a really low metabolic rate, and they eventually terminate diapause and initiate adult development. And we could look for transcriptional regulation differences, at least through microarrays, between these different classes or categories of flies. And we could see that a whole bunch of genes are up and down regulated during this sort of critical decision-making period. In fact, it looks like diapausing pupae act like they're really hypoxic, as if they're going into oxygen deprivation. And a lot of the pathways are related to this. We can also look at diapause termination and do the same kind of characterization of metabolic rates as an individual pupae comes out of diapause and then begins adult development. We see this plateauing where there's an initial slight rise, relatively plateaued, non-change in metabolic rate, and then an uptick again as the fly completes adult development. And we can look and have looked at expression patterns through all these different stages of diapause termination and subsequent adult development. And we've looked concentrated a lot on this initial period right here. And eventually what we found is that we've now got 18 candidate loci that are really strongly up or down regulated right at what we think is the window or Q window when diapause termination is taking place. Some involved in wet signaling, tyrosine metabolism, some biosynthesis of steroids. There are a set of particular traits that we might predict could be involved in dipause termination. We've also seen that at 24 to 48 hours, there's also a concerted changes in expression patterns of a lot of different genes. So there may be another critical period for timing, or besides just breakage itself, or some of the key genes that are up and down regulated during diapause breakage. So in this respect, what I'm trying to say, or on potential, is that even though there may be key or critical players or individual loci that are involved in the specific signal or target for terminating diapause, with a cascade of subsequent genes being regulated off of that key sort of keystone locus, it's also possible that there's a lot of interplay between the pathways and determination of when that initial signal occurs, and downstream there may be real important reasons for concerted patterns and related patterns of differentiation of genes in the downstream pathways, such that a number of loci, when one's selecting for a different life history profile, could be under selection. It's not just as simple as thinking, oh, diapause termination is different between these two flies. It just must be one master control timing gene, and that's all that has to change to shift the life history. What I'm arguing is it might actually be more complex than that. And it could involve a coordination of a number of different genes, not just that one trigger. And so what I want to leave you with is a study that was done by Molly Burke, Tony Long's lab, that just recently got published in Nature, in which this selection was performed on Drosophila melanogaster, so we'll return to a model system. And selection was performed for shorter developmental timing in the fly. Somewhat similar to what we might expect, for instance, in the apple race, with a more rapid rate of initial adult development and earlier diapause termination time. Right? And what these investigators found, in fact, in a number of replicated lines, 2 replicated lines that were selected long-term for faster larval development to adulthood, they found, looking at genome scans, defined associated regions that differentiated between control lines and these selected lines, they found evidence for widespread response across the genome of Drosophila melanogaster. It wasn't just a few loci. There were a number of loci that showed a response, and many of these loci fit with what one might expect when one's selecting for faster rates of development. There are many signaling pathways. Some were involved in wing disc development, others in metamorphosis and organ development, imaginal disc morphogenesis, etc. So the response here, at least for what I'm saying, is for a different life history profile, was quite dramatic across the genome, which fits with at least an argument that maybe selection for life history differentiation in Phytophagous insects and adapting to host plants with differing phenologies, different trimming times, could actually result in widespread divergent selection across the genome of the flies. So to now link together what we see in terms of the genomic surveys of these flies, right, with patterns that we see from selection experiments, with patterns that we see in the transcriptome analysis. So we're making a case that maybe in the early stages of divergence, and at least in Rhagoletis and other Phytophagous insects that undergo host shifting, when that involves a life history shift, the selection could be widespread across the genome, and the genetic response could be more widespread than what we thought previously. It may not be just islands of speciation. It may really be that we might see continents of divergence instead. There'll still be a topology to those continents, depending on selection strain, recombination rates, etc. But the whole of the genome might be more elevated above that neutral sea level. And so including then this high throughput sequencing might really shift our focus to thinking about questions about the genomic architecture of divergence, away from just what has been a fruitful sort of beanbag approach looking at individual speciation genes. We can now begin to think and ask questions and actually focus attention on genome-wide patterns of differentiation. And it might modify our view of the genetics of speciation, beginning to allow us to place individual speciation genes at least to be studied as part of a collective and evolving genome. So thank you.
So I think maybe if you have questions, you could join us for lunch. So right now we're gonna head over to the silo for lunch there. I think we ran a little bit over, so I think that would be best if you have questions to join us there or come up. Thanks everyone, and thank you very much. Okay. All right, everybody, so let's get started. Welcome to the final seminar of the quarter for entomology. We're ending with a bang today with Dr. Jeffrey Fader. He got his PhD at Michigan State University working with Guy Bush. He then had postdocs at Princeton and the University of Chicago. Following that, he got a faculty position at the University of Notre Dame, where he is currently a professor in biological sciences. His lab there works on speciation and host specialization in the Rhagoletis pomonella species sibling complex. And they use field work and selection experiments and molecular genetics to answer all sorts of interesting questions in this area. They also are looking at color polymorphisms in a bioluminescent Jamaican click beetle. He's also the director of the GLOBES program there, the Global Linkages of Biology, Environment, and Society. So without further ado, I'll get started.
Oh, thanks. Can everybody hear me in the back? It's fine. Good. Thanks, Ian. Thanks, Meredith, for inviting me. It's been a lot of fun so far. Thanks, you all, for being here today. And I'm going to tell a story about genomic architecture of speciation today. And of course, speciation is the ultimate source of biodiversity, and so this is sort of the big issue, at least in putting the talk today in the context. We're trying to understand speciation and biodiversity, but specifically I want to sort of concentrate on the question of what's the nature and role that genome structure itself might play in speciation. And I'm particularly interested in this question with respect to taxa or populations that are diverging in the face of gene flow or migration between the populations. That is, let's say, in a geographic, and that low level of neutrality that we set for statistical purposes may actually be too high, and we're missing a number of regions of the genome might not be under as strong a selection, but are still under divergent selection during these early stages of population divergence. And that's the take-home message that I want to impart, and that's what's shown in this figure here. Rather than this view of sort of genomic architecture of divergence in these host races of Rhagoletis flies, we see something that looks more akin to sort of a large uplifting and more widespread differentiation. Okay, so let's sort of take a step back and sort of lay what I'm talking about today in a larger context. And that I'm saying, well, what often initiates speciation? And many times it could be that reproductive isolation due to divergent selection, adapting populations to different habitats, is an initial impetus or driver of the differentiation process. And so what I'm saying here then is that you could think about it in the context, let's say, of fitness trade-offs. maybe we have two habitats, and you can see the association here with Apple versus Hawthorne for Rhagoletis pomonella, the host that attacked. Maybe we have two habitats here. One environment, like a desert environment, the other environment, maybe an alpine meadow environment, right? And what we've got is differential selection pressures on traits to adapt the population to those different habitats, such that maybe a gene or an allele that's favored in producing a phenotype that confers higher fitness, let's say, in our desert habitat, has the opposite effect and results in a phenotype of lower fitness in the alternative habitat, and vice versa. So this is our context for divergent ecological selection. And if this is the case, then we'd expect sort of to see a heterogeneous pattern of genetic differentiation throughout the genome. So what do I mean by context of what we call parapatry, partially geographic overlap of population, or sympatry, that is, would be more complete geographic overlap of populations. Now, we've learned quite a bit about the genetics of speciation over the last couple of decades. And particularly with sort of an emphasis on identifying individual genes that are contributing to reproductive isolation. We could call this sort of the hunt for speciation genes. And this has been quite informative about telling us about the genetic basis of reproductive isolation that can be contributing to speciation. There's a couple of difficulties here, though. Many of these genes that have been identified so far affect post-zygotic incompatibilities, and a lot of times these crosses are done to investigate the genetics of speciation through sort of long established and somewhat highly diverged taxa. And so some of these loci may have actually diverged after speciation was essentially complete. And also, more importantly for the talk today, the hunt for the speciation genes, it's been quite informative, but it also misses another issue, and that is thinking about how the genes themselves that are diverging are arrayed together in genomes and might diverge collectively and might influence further differentiation during the formative stages of speciation. And so today we're going to concentrate on this fly here as our main character, Rhagoletis pomonella. Rhagoletis pomonella, of course, is a model system for ecological speciation with gene flow in phytophagus or host-specific phytophagus plant-eating insects. The basic premise for Rhagoletis pomonella is that in the process of shifting and adapting ecologically to new host plants, this provides an impetus or some of the main factors that are driving the divergence process. And the particular example of Rhagoletis pomonella by that, well, what I mean is that for genes that are directly under divergent selection between the two habitats, producing a phenotype that's directly selected for differentially, we'd expect to see if there's some gene flow going on between the populations. For these traits that are directly the targets of selection or nucleotide sites, we'd expect to see strong differentiation between our populations. So these regions of the genome, if we were to scan through, should show high levels of difference between our populations. We could also have genes that aren't directly under selection, or sites that aren't directly under selection, but are physically linked close by. And those sites should also show a degree of differentiation between our populations, but not as strong as those sites that are directly under selection. And then finally, we should see sites that aren't directly under selection again, neutral, at least in terms of fitness effects, but unlinked, that is, at greater recombination distances to a locus directly under selection. And in this case, these genes should flow more freely between our populations that are divergently selected, and we don't expect then to see high levels of genetic difference between our populations if we were to score individuals at this locus and genotype them. So far, so good? Okay, so we expect a heterogeneous pattern of genomic differentiation, which sort of has led to a genomics island of speciation sort of analogy. And that is, we might expect to see, initially during the speciation process of speciation with gene flow, certain regions, just a few, scattered through the genome that are under high divergent selection that show differentiation between our populations. And then by a process that Saravia has called divergence hitchhiking, we'd expect to see then reduced levels of gene flow in a shifting from its ancestral host, hawthorn or thorn apples, to introduce domesticated apples about 150 years ago in the eastern United States is often cited as an example of this type of sympatric or ecological speciation with gene flow in action in our own backyards. So this provides us with at least a known geography, sympatric, geographically overlapping, and a historical time frame, then about the last 150 years ago, the shift occurred, that allows us to sort of investigate or sets up the preconditions, at least, for doing an investigation of genomic patterns of differentiation underlying speciation with gene flow in the very early incipient stages. And the take-home message I want to impart on you today, what we're going to do is we're going to integrate geographic surveys of genetic differentiation across sites with selection experiments and mapping experiments. And the take-home message is that surprisingly, or somewhat unexpectedly, at least in my opinion, what we seem to find are large continents of differentiation across the genome rather than small sort of isolated islands or genomic islands of speciation. A different genomic architecture, at least underlying the incipient stages of host race formation in this fly than I would have thought. And what we're going to, one of the sort of implications of this is that maybe these, and I'll explain this a little bit more fully as we go, some of the low baseline levels of differentiation that we detect in genome scans. And by that, a genome scan, what I mean is looking at populations and scanning a number of loci across the genome of the fly or organisms to characterize genetic differentiation on a sort of more genome-wide scale. Many of these studies sort of set a baseline level above what we'd expect statistically for neutrality. In many cases, actually, selection may be more genome-wide and widespread, right?